doing regular videos. Be on the bus. Those are the last three of those videos with questions, experimental videos. We do have one more video, which is really in the lab category, which is the reproductive video. That's not due till like Debbie, but that's the last lab. That one, there's not questions on there. You watch the video and you do a one-page outline, just bullet points, and you upload that, and that's the last lab report. So there's that. Video. That's on Canvas. You can see it there. So, in the somewhat distant future, there is a book report. And I think that's due during Fed Week as well. But I know a lot of you don't like to wait till the last minute. And so that book is The Man Who Mistook His Wife for a Hat and Other Clinical Tales by Oliver Sacks. And so basically, you just need to read that book and write about a two-page typed report on it. A book report, essentially. That book report, just to give you a little bit of guidance, you're not really looking for a summary as much as an evaluation or a reaction. You will probably need to add some summary in there to help you make your point. But we're mostly wanting you to focus on your reaction or evaluation to the book. Because your book report is only 2 pages long, what I would suggest is read the whole book, but it's separated into 4 subsections, and you might want to focus your book report on one of those subsections, or maybe even some chapters within a subsection. That would be fine. The idea is to read the book. I think you'll find it very interesting. Has anybody read it before? Yeah, at least one. A lot of times somebody's read it somewhere along the way. So I think you'll find it interesting. But read that book, and then remember we have about a 2-page book report. It's listed, the due date in your syllabus and online and all that. But you can just upload the book report to Canvas. So anyway, that's it's still a ways away, but things get busy towards the end of the quarter. But I just wanted to sort of have that in your mind. That's out there too on the to-do list eventually. Any questions about book reports or anything else that you want to chat about? Okay. Yeah.
Will there be a rubric for the book report that we can access?
On Canvas, there is some instructions about how to do the book report. There's no official rubric. I think essentially, obviously, we want it as well written as you can. If you quote from the book, put it in quotation marks or something like that. That kind of standard. We want it to be your original work for the book report. Like to have you keep it to about 2 pages. If you go a little over, that's no big deal, but we don't want 10 pages. It's got to try to keep it tight. And again, you can narrow in on a certain portion of the book. That's fine. Usually, you have an introduction, and then you talk about your main thing, and something like that. But rigid. You can, use some of your creativity there, but, it's a lot of it is your reaction and, critique of the book, if you will. And a critique doesn't necessarily, it's gonna be negative or, you know, positive, it could be either one, or just your thoughts on the book, reaction to it. And again, you can put some summary in there, I don't wanna hamper you too much on that, but what I'm not really looking for is, this chapter was about this, okay, this chapter was about this, I mean, if, in making your point, you may need to do that for a couple chapters, but then do a reaction to that, not just a quick summary of each chapter, that's not what we're looking for. I don't know if that helps, but, that's kind of the idea. And when you get into it, if you have some more specific questions, you can the main reason I want it to be a well-written book report, but the main reason I assign it is because, I think you'll find reading that book valuable, and the main idea is I want to give you credit for reading the book, and, so that's the idea of it. All right, well, let's go back into the vision that we've been introduced last time. If we look at some more of the issues here, we see that we're reminded that last time we talked a bit about refraction, and that there are 4 major parts of the eye that are involved in refraction. The main structure that refracts the most, bends the light the most, is the cornea, where it comes into the eye, that transparent cornea at the anterior part of the eye. Then there's a little bit of refraction with the fluid, the aqueous humor, and then the lens has a modifiable refraction. You can change it with the changing of the shape of the lens. And then the vitreous humor, a little bit of refraction, and so those are the 4. Now, we're trying to focus the light right on the retina, and so that's the goal with these refractive structures. If it's done very well, then we have a condition called emmetropia, this guy right here. And that's where this little very simplistic diagram of the eye, there's the lens there in yellow, here's the cornea. It gets focused right on the retina. There's no correction needed. Everything's wonderful, all right? Then under B here, we have myopia. Myopia is the most common type of eye problem in younger folks. And what happens here is the cornea is too long, and so refraction is too great, and it comes to a focal point in front of the retina instead of right on the retina. To fix this, we could use a concave lens to spread out the light rays, and of course you go to the eye doctor, as many of you've done, and said, okay, is this one clear, or is that one clear? So they pick the right power for the lens, and then they set it up, and you have good vision with your lens. This could be glasses, or it could be contact lenses, but it's fixing it in that way. Hyperopia, or farsightedness, of course, myopia, that means you can see near, close up. It's also called nearsightedness. You don't see well far away. Hyperopia, or farsightedness, means you can see farther away and not close up. In this case, the cornea can be too short, and so the light rays don't come to a focal point even by the time they hit the retina. So they would come to the focal point if they were allowed to behind the retina. Of course, the retina blocks it. to correct this, we would use a convex lens and to pull it back tight onto the retina. Astigmatism is your cornea is uneven, and so it gives a uneven image there on your retina, and so you'd have to correct it with an uneven lens to help correct that. A lot of people that have myopia have at least some astigmatism as well, so that's fairly common. Now there is another procedure, mostly for myopia. Because the cornea is too long, you can do a laser surgery, a LASIK surgery, and cut the cornea down and trim it down so you don't have to wear glasses. There is some value in this and some downside. It's not recommended in people under 25 because their eyes are still changing quite a bit. After that, it can happen, but there are some possible side effects to it and risk factors. But that's the idea of correcting it with a laser. So those are some of the problems. What about some other eye disorders? All of these here are issues with the cornea. And so we put that in big letters up here, issues with the cornea. Okay? What about others? Well, cataract and presbyopia are issues with the lens. Cataract is, as we age, the light rays have energy, and over time, the lens becomes damaged to some degree, and it becomes more cloudy, more opaque. The lens, like the cornea, needs to be transparent. It needs to allow the light through on its way to the retina. If as damage occurs over time, it loses its transparency and it becomes more opaque. You can't let the light come through. And so that is a cataract. How do we cure a cataract? Usually you take the lens out. You mostly now these days put an artificial lens in. There is an option of just leaving no lens, but that affects your accommodation, of course. But usually they put an artificial lens in that is, of course, transparent. And so cataract surgery is one of the more common, probably the most common eye surgery. And often people get cataract surgery in their 70s or 80s or beyond. But usually it's often difficult to go too much past in the early 80s without having a cataract surgery. Of course, we see this in aging animals too, dogs and cats and whatever. You can see that cloudiness in their eye. You can see the cataract. If it's not treated, eventually it could lead to blindness, but usually in humans that do a treatment before, if possible, before that happens. Presbyopia we talked about a little bit. The lens becomes less flexible with age. The lens is supposed to change shape for accommodation, and it doesn't do that as well, so we can't accommodate as well, which means we cannot see near vision. It's harder to read, look at the phone or whatever device, because so you try to put it way out here, you can see a little better, or you wear reading glasses, something like that. And generally that's the only treatment you're going to do, is wear some kind of glasses to help you see in the close-up better. If the rest of your vision is okay, you would just use reading glasses, which sort of magnify that close-up range. Preterm birth affects the retina. So this one here is a retina. Preterm birth is defined as less than 37 weeks gestation. Gestation can be, we sort of have to define that a little bit more precisely. In humans, the gestational period is usually what we would technically say 38 weeks. It takes 38 weeks from conception to delivery. But for convenience, we usually back that off about 2 weeks because we don't know precisely when conception happened. We back it off to the last period. So you go 40 weeks and you add 2 weeks to that. So if that's the case, then you're looking at a 40-week gestational period with starting 2 weeks earlier than conception approximately. So by that definition, a preterm birth out of a 40-week time would be about 37 weeks. The danger is increasingly great the earlier the baby is born. And certainly anything before 31 weeks or a weight of less than 3 pounds, the birth weight of the baby, makes it a much higher risk. What is the risk? Well, the retina develops, where our photoreceptors and other neuronal cells are, develops fairly late. It starts to develop in the second trimester. We can also call the gestational period, we can separate it into trimesters. The first 3 months is first trimester, second 3 months, second trimester, and the third 3 months, third trimester. Well, we can separate it in that way, and that will give us some indication. So the eye development in the retina begins in the second trimester, but again, if birth is too early, it hasn't had a chance to fully develop. And so the retina may not be developed appropriately. Now, the lungs develop pretty late too, and surfactant and all of that. And so a lot of times, if you have a preterm baby, especially earlier, you need to give them oxygen because their lungs are not fully developed. So you're giving them a higher amount of oxygen, maybe have them on a ventilator or something like that. The problem is the retina, if you have higher levels of oxygen, the retina produces blood vessels that are not quite normal. They're a little abnormal, and you can have an overgrowth of them too. And then if you cut it, then the body produces a growth factor, and then you end up getting, again, somewhat potentially damaged, or at least not normal blood vessels, and you can have some overgrowth. And this can lead to blindness or at least a vision deficiency. So blindness or at least a decrease in visual defects can happen with preterm births, and the earlier it is, the worse. And then you have to worry about how much oxygen they're giving them. They need oxygen to develop everything, but it can cause problems with the retina. So that's a problem there. So that's a retinal problem. Another retinal problem that isn't on the list here, but if you have a detached retina, usually if you get a blow, you fall down, and something hits you, it can knock your retina off the back of the rest of your eye, or partially off, and you can try to reattach it, but it may work or may not, or it may partially work. And of course, if it's detached from the underlying vascular blood vessel supply, then you can have some death of your photoreceptors and neuronal cells, and this can cause some blindness and or loss of vision. So that's another one that we could very well, probably should add here. Glaucoma, we have a circulating fluid, the aqueous humor, in the anterior cavity, the anterior posterior chambers, and it drains through what's called the channel of Schlemm, and so you produce it and drain it, And anything that increases the pressure inside the eye, intraocular pressure, is classified as glaucoma. It can be a blockage of the fluid, or it can be due to other things, and that could damage your optic nerve, cause other kinds of damage. And glaucoma is one of the risk factors associated with diabetes. The diabetes, if the sugar level isn't kept in close enough homeostasis, you can get increased pressure inside your eye. And it can also, diabetes, or at least the lack of keeping your sugar in balance, can damage the endothelial lining of your blood vessels. We mentioned before that that can cause a decreased circulation to the lower extremities, which might cause amputation eventually of the feet or what have you. But it can also cause problems with the blood vessels in the retina. We do have blood vessels in the retina. There's not as many because you don't want to block the light. And you have the choroid layer underneath, which is very vascular, and you can have diffusion of oxygen and nutrients from the choroid layer. But we do have some from in the retina, and if we damage those vessels, the lining of them, we can get blindness or visual deficiency due to things like diabetes type 2. That's a risk factor for it. And so some people who have diabetes become blind or have visual impairment. Macular degeneration, we have another slide about this. It's a little bit more extensive, but basically we have the macula lutea is the yellow spot right in the center axis of the eye. And that's where we have our greatest visual acuity. And if you get a loss of vision there, it makes a sort of a black spot in the middle of your eye. You can see peripherally, but that's not as good as acuity. You have your greatest concentration of cones in the middle axis of the eye. And macular degeneration, age is a risk factor. A lot of people get it as they get older. Genetic predisposition and smoking. For older adults, macular degeneration is the most common cause of blindness or visual deficiency, maybe it's a better way of saying it. Well, here's our retina. If we look at this, there's some oddities here. The front of the eye is over here, okay, where the arrow is, and the light is coming this way. So here's the light coming this way. But your photoreceptors are on the back of the retina, the posterior part of the retina. Logically, intuitively, we would probably say, why aren't the photoreceptors to the front of the retina? Why are they on the back? Well, we don't know for sure, but one hypothesis that seems fairly logical is having to do with getting the oxygen and nutrients. The retina, again, although it does have blood vessels, it doesn't have as many because it could block the light. But this underlying choroid layer, right underneath the retina, is very vascular, and so the very metabolic active photoreceptors in this design are closest to the choroid layer, so oxygen and nutrients can diffuse a much shorter distance than if they were at the front of the retina. So that's a theory anyway. Our two photoreceptors are the rods and the cones, and it's sort of based their name on the shape. The cones have a more conical shape, and the rods have a more rod-like shape. In both cases, they're going to respond to changes from dark to light. We're going to see in both the rods and the cones, there's an outer segment, this portion out here, and then there's an inner segment where you have the nucleus and mitochondria and a lot of those kinds of things. In this outer segment, you have a series of membranes that are lined up kind of like plates. These are electron micrographs of the cones and the rods. these membranes are going to have photopigments within them. These photopigments are going to be able to absorb light, and so they're absorbing light. Just like a pigment in paint can absorb light, these photopigments absorb light, and they're going to yield changes inside of the photoreceptor cell, metabolic changes, opening and closing ion gates, things of that nature, which can lead to changes in the polarity. Essentially, one odd thing, again, with vision is the photoreceptors are depolarized in the dark, in the lack of a light stimulus. And when light hits them, you have a relative hyperpolarization. We're going to talk quite a bit about that today, but all the other sensations that we're aware of, the stimulus causes a depolarization, but not for the photoreceptors. They have a depolarization in the dark, no light stimulus, and they're hyperpolarized in the light. We call that depolarization in the dark current, by the way. Now these photoreceptors impinge directly on bipolar cells. They're called bipolar cells because they have this bipolar configuration. And the bipolar cells will then impinge on ganglion cells. It's interesting to note that there's only local current in both the photoreceptors and the bipolar cells. No action potentials. We get depolarizations and hyperpolarizations in the photoreceptors and the bipolar cells, and we don't get action potentials in this direct visual pathway until we get to the ganglion cells. The ganglion cells will generate action potentials, and they'll swing around with their processes, their axons, and it'll swing around and go backwards out the back of the eye, and that generates our optic nerve. Where the optic nerve exits the back of the eye, it's called the optic disc, or the blind spot that we mentioned briefly. We have a couple of other types of cells in the retina. We have some horizontal cells and amacrine cells. We're not really going to talk about those much, other than to say that they help with some integration of the signal, but they're not in the direct signal pathway. So we're going to focus mostly on the direct signal pathway. This is a little bit bigger image of it. So we have synapses between the photoreceptors and the bipolar cells, and then we have synapses between the bipolar cells and the ganglion cells. The neurotransmitter that's released from the photoreceptors at this synapse, actually in both synapses, is glutamate. Glutamate can bind to glutamate receptors on your bipolar cells. The fascinating thing here is we have two subclasses of bipolar cells. One subclass, the binding of glutamate causes depolarization. The other subclass, the binding of the glutamate causes hyperpolarization. You open different gates. On and off. These are on and off centric pathways. And they are adjacent so that it provides some contrast. Before we get back to the on and off center and those responses of glutamate, let's go to the basic visual transduction mechanism in the photoreceptors. within the photoreceptors, in their outer segment, we mentioned they have some membranous disc, some membranes lined up. And within those membranes, there are photoreceptors, photopigments, excuse me. The whole cell is a photoreceptor. There's photopigments. The photopigment in the rods is rhodopsin. The photopigment in the cones is also an opsin, but it's usually scrotopsin of 3 different varieties. This may be a good place to point out, we have 3 different versions of the cones, each having a different opsin, where we have only 1 version of the rods. We need 3 different versions of the cones because they absorb light in slightly different wavelengths, and they're the ones that are then going to perceive color. The rods only perceive black and white. Well, with that having been said, when the light hits an opsin, which is a protein in one of these disc membranes, it can cause a chemical reaction, and in rods this is called retinal. Retinal is a derivative of vitamin A, and this is why we often say, oh, you need to make sure you have enough vitamin A if you want to see well. Well, this is a derivative. So retinal, we're going to say retinal, or retinine as it can also be called, can exist in 2 different conformations. Attached to this opsin retinal complex is a G protein called transducin. And transducin can be activated by light and convert guanosine triphosphate to guanosine diphosphate and activate a metabolic pathway that we'll be looking at very shortly. When we look at the conformational change of retinine, here's our opsin, here's 11-cis retinine, which is kind of bent shape, and here's the all-trans retinine. You don't need to memorize this chemical structure, but you can see that there's a conformational change. This change occurs when we go from dark to light. When light hits, the 11-cis retinine is converted to the all-trans form. And that is then going to activate transducin, this G protein, and cause a change in our guanosine tridiphosphate concentration. Okay. Before we look at an important diagram in understanding this, remember that ganglion cells in our direct pathway are the only ones that produce action potentials. Photoreceptors and bipolar cells only produce local currents, depolarizations and hyperpolarizations. Now, amacrine cells, which are sort of helper cells there, also can produce action potentials, but again, we're not going to go into that in too great detail. In the dark, photoreceptors are depolarized and they release the neurotransmitter glutamate. This neurotransmitter glutamate can either hyperpolarize or depolarize bipolar neurons, depending on their type, depending on the type of glutamate receptor that they have and which ion gates they open. Light inhibits the release of glutamate, so it decreases glutamate release. In the dark, the rods and cones have many sodium and calcium channels, and they're open, and that causes a depolarization. Now, this part here, we're going to look at it on a diagram, but it's all listed out here. So I'm not really skipping this page, but I want to show it. It's easier to understand if we jump to the diagram. We should know this diagram well and what's happening here. First of all, we see with the blue arrows, these are the processes in the dark, and in the orange arrow, these are the processes activated by light. Here's our photoreceptor. Now, in the dark, we have created a number of cyclic GMP molecules. This is due to an enzyme, guanonyl cyclase, converting GTP to cyclic AMP. The cyclic AMP causes sodium and calcium channels to be opened in the membrane. Sodium and calcium will go down their concentration gradient into the interior of the photoreceptor. This will cause that dark current, that depolarization in the dark. So that's the initial condition. Now as we swing over here, the light, when it hits the opsin, the photopigment, and is absorbed, will change the retinal, in this case, from 11 cis to all trans. That will activate a G protein, which is just named transducin in the visual transduction process. It's a G protein. This G protein, or transducin, will activate an enzyme called cyclic AMP phosphodiesterase. What that enzyme will do is it takes cyclic GMP and it converts it to 5' GMP. That's what this is over here, 5' GMP. Essentially what you're doing is you're removing cyclic GMP, you're converting it to something else. And so as your concentrations of cyclic GMP decrease, you close your sodium and calcium gates. You no longer have the cyclic GMP to hold them open, so they're closing. So sodium and calcium cannot diffuse in. This creates a relative hyperpolarization compared to the conditions that existed in the dark. Are we all still okay? Are we like, oh no, we jumped off a cliff here, where are we going? You feel okay so far? Okay, this is the pathway. This is what's happening. All right, so that's the light reaction. So this is listed here in the previous page in text, but again, I think it's a lot easier if we have something visual, this graphic to look at while we're chatting about it. Okay, so photoreceptors depolarized in the dark, the light hyperpolarized in the dark. Now we're back a bit to these pathways, on and off pathways. We have light hitting in different patterns on the retina, and you have on and off pathways when you consider your ganglion cells, how the pathway of the photoreceptor to the bipolar cell to the ganglion cell, how it reacts. So let's go through this a bit. We'll have quite a bit of text explaining this, but looking at something like this, I think is perhaps the easiest way to understand it. We have an on pathway and an off pathway. The first three steps are the same whether you have an on pathway or an off pathway. After that, they're opposite effects. Notice right here on this step, this is where the light is hitting. The first step is the photoreceptor is depolarized in the absence of light, and that's true for both pathways. Now when light hits, that causes a hyperpolarization. It closes those sodium and calcium gates by that mechanism of converting cyclic GMP to 5' GMP. By the way, not to go off track here, but eventually this 5' GMP, it can get reconverted through the GTP back to cyclic GMP. Okay, there's a metabolic cycle in there. Okay, let's not get too distracted by that. Okay, the light hyperpolarizes in both pathways by shutting those sodium and calcium gates. Now, that will cause in both pathways this hyperpolarization less glutamate released, because the glutamate is released due to the depolarization. The hyperpolarization causes less glutamate to be released. Well, the light causes less glutamate to be released in both pathways. Well, in the on pathway, glutamate, if it's there, inhibits the bipolar cells, causing hyperpolarization and less action potentials in the ganglion cells. So if you have decreased glutamate, you reduce your inhibition and in fact depolarize your bipolar cells and lead to more action potentials. Ooh, did we wrap our brain around that? Let's say it again. In the on pathway, these subclass of bipolar cells, if you bind glutamate to them, they will hyperpolarize. They'll be inhibited. So, if you have decreased glutamate, which happens during the light, you will reduce that inhibition because you have less glutamate. And so you will reduce inhibition means more excitation. You'll have more depolarization and you'll get more action potential in the ganglion cell. Just pausing to let that sink in for you. It's a little bit of a, like a double negative. Now, in your off pathways, what's different? The first three steps are the same, but your bipolar cells, when you have more glutamate, they would inhibit your bipolar cells, causing them to be more hyperpolarized, and you'd have fewer action potentials. So when you get light hitting on this pathway, you will have decreased glutamate released, and in that case, you'll reduce your excitation, not your inhibition. And so you will hyperpolarize these cells, and you will then get fewer action potentials. These pathways tend to be adjacent to each other, so you get contrast. So that some pathways that are going by the optic nerve and eventually to the occipital lobe, some of them are having more action potentials and some are having fewer action potentials as you're having light and dark, or light and less light hitting these pathways. And so you can get contrast, you can see edges, you can see shapes a little more. So it's giving you that contrast ability with having this opposite reactivity. This shows these light hitting these different areas. To me, the easiest way to understand it is to understand this flow chart. Now, the explanation for this is gone into in some detail here. This is what we've just talked about, but to give you hope, there's an explanation here as well, and it's also outlined in the book. And you can see here's the two key differences. First of all, the photoreceptors are depolarized in the dark. The light hyperpolarizes them, causing less glutamate release, but you have two different bipolar cells, and they'll react in an opposite way to glutamate. So a reduction of glutamate will cause different aspects. So this is the explanation that we just went through, but it's laid out for you in these couple of pages there. In addition, though, to that discussion, a subset of ganglion cells also have an opsin-like pigment called melanopsin. This goes to a part of our hypothalamus called the suprachiasmatic nucleus, and that absorption of light by melanopsin in the ganglion cells sends some signals to that suprachiasmatic nucleus, which helps entrain our internal clock, our biological clock. So that helps in training. So that's nothing to do with the visual pathway, it's in training our biological clock. We also have some other pathways. Instead of going to the occipital lobe of the cerebrum, some goes to the brainstem and the cerebellum, which helps coordinate our eye movements and head movements and all of that. Rods are very sensitive to light, but they're not very good at visual acuity. Rods tend to be a little bit more on the periphery of the retina, and so in dark or dim conditions, we actually see a little bit better out of the corner of our eye instead of looking straight out at an object, because we want to put the light on the periphery of the retina, the rods. So rods are good for night vision or dim, low light conditions. Cones need more light, but if they have adequate light, they have more visual acuity. There's also 3 subtypes of cones, and so they give us color vision. These are the 3 subtypes of cones. One subtype, the opsin, the protein, absorbs mostly at around 420 nanometers, around blue light mainly. One absorbs more in green light, and one absorbs more in red light. Rods tend to absorb just black and white. They don't see color. There's only one type. Because we have absorption of 3 different parts of the spectra, the brain can then take these 3 primary colors, if you will, blue, green, and red, and integrate them into all the colors that we see. Because these are proteins, these opsins, if we have a mutation coding for any of these proteins, then we will have a misshaped opsin and a less effective absorption, and we can have color deficiency. Hardly anybody's actually totally colorblind, but you can have a color deficiency. Because the most common of these opsins, the red and the green, have their genes coding for that opsin on the X chromosome. Males have a much greater chance of color deficiency than females, because as you know, women have 2 variants of the X chromosome and men only have 1. So a woman could be a carrier without actually having color deficiency. To have a color deficiency, she would have deficiency in both alleles of her X chromosome at that gene. Whereas men, they only have the 1 variant of the 1 allele of the X chromosome there at that gene, and so they would have more common color blindness. Before we leave, if you don't mind, let's do a quick little experiment, and we'll finish up the rest on Monday, but I'll look at this for you, with you. If you can, let's stare at that yellow with the triangle in it, and just keep staring at it. Eventually I'm going to say something to now or something, and you'll move it to your vision to the right where the square is. But right now, just keep staring at the yellow and the triangle. And there's a reason why we keep staring at it. And I'm sort of stalling because we have to stare at it for quite a while here. Okay, now get ready. As we're staring at this, I'm going to say now in just a bit, and then switch your vision over to the right where the square is. Okay, are you ready? Now,
Wow.
what do you see?
Oh,
Circle.
You saw bluish purplish, right? Isn't that freaky?
That's creepy.
You know why?
Brandon's lazy.
Taking some weird drugs? No. We have these three options, right? This is white over here, what we looked at. We fatigue our red and green option because that makes yellow.
Oh.
We fatigue these two, and when we looked, what ones weren't fatigued? Blue. So bluish purplish is what we see when we look at the white, because we fatigued our others. Isn't that fun? Okay, that's what we did. On Monday, we already talked about color lines, so we'll do these last 2 or 3 pages before we finish up the chapter there, and then we'll move to the muscles. Okay, have a wonderful weekend. Sorry to feel bad. I thought you'd enjoy that last one.